Your sound is off. So I'm not hearing anything. Okay, can someone confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? Let me see if we're on chapter 6. I'm not sure we're on chapter 6. All right, I'm not hearing anything, so let's begin. Let me share my screen here. Oop, that's the wrong one. All right, that was a little long and difficult to do, but I just shared my screen there. Nothing really to see. Let's go back to the home. Let's go to the syllabus. So today is August 7th, and we should be on chapter 6. Is today Tuesday? Thursday. And we should be finishing chapter 6 and beginning chapter 7. There is a lab today, lab 8 in the lab today at 7. Any questions about what we'll be doing? Let's go to chapter 6. And I've got that we were talking about agar, and we discussed that agar is something that's obtained from seaweed, a polysaccharide, and we use it in our media to solidify solid media, meaning make liquid broth a solid media. And agar is something that actually bakers and cooks have been using for a long time. I don't know, hundreds of years, something like that. And I can mention that Dr. Koch didn't like the fact that his agar plates, and he was using gelatin at the time, sometimes the bacteria dissolved the gelatin, in which case he had a liquid mess in his agar plate. And he didn't like that. And it was actually the wife of one of his assistants who suggested he try using agar. And he did, and he really liked it. And we still use agar today because bacteria cannot digest it. Any questions about agar? All right, we were talking a little bit about media. There are different types of media, as you well should be aware of. There are chemically defined media, and these media have the exact chemical composition known because all of the molecules added to the media are something that somebody added. Let me blow this up and then I can show you a simple one. A simple media, glucose salts media, having glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and then water. And these are the only molecules that are added to the media. This is a chemically defined media, and we know what all of the chemicals are in the media. Now, a fastidious organism could never grow in this because it doesn't have the additives required. And this is a media. Oops, let's go back. I have to blow it up here so you can actually see it. And this is a media that somebody has made for a fastidious organism. And besides glucose and azide and citrate and oxaloacetate and all of these, I don't know, salts of different chemicals, potassium sulfate, sodium sulfate, magnesium chloride, giving all these things that an organism might need. It also has different growth factors like cysteine, arginine, glutamic acid, asparagine, cysteine, thiamine, a B vitamin, nicotinamide, adenine dinucleotide, that's NADH, Uracil. I used to know what that was. A biotin. I used to know what that was too. That might be a B vitamin. And then uh, hypoxanthine, and then sodium thioglycolate. So all of these are a chemically defined medium, but you can see that we're adding a whole bunch of different ingredients because this organism is a fastidious organism and it would not grow on a simple media. Chemically defined media are normally only used for autotrophs because they don't require a lot of additives and it has glucose in it, so that's supplying both a carbon source as well as an energy source. Any question about any of that? So for most bacteria, we use a complex media to get them to grow. Like E. coli, E. coli might grow in glucose salts media, but it definitely will grow in a complex media. And this would be something like tryptic soy agar or nutrient agar Na. This contains extracts and digests of yeast or meat or plants, and these provide both vitamins and organic growth factors, but the exact chemical composition of a complex media is not known. For example, let me blow this up. Here's a nutrient agar, a complex media that has peptone. This is a partially digested proteins that we obtain from soybeans, and then beef extract, 3 grams of it, sodium chloride, 8 grams, agar to make it a solid media, and then water. And this is a very simple media, nutrient agar, and this will grow E. coli. But we don't know all of the different molecules because it'll contain all of the different molecules from the cow. Whatever the cow was eating, whatever molecules were in the cow will be in this nutrient agar, the complex media. And we don't know what the cow was eating, so we don't know what all the molecules would be. Any question about any of that? Peptone, the partial protein digest, is both a primary energy source, supplying energy for the microbe to digest, as well as a carbon source, a nitrogen source, and a sulfur source. Complex media are normally used to grow heterotrophs, bacteria and fungi, that require an organic carbon source. Any question about any of that? We have different types of culture media, such as anaerobic media. This would be important for an obligate anaerobe, one that must avoid exposure to oxygen, because oxygen would kill it. An anaerobic media has a reducing agent in it, and that's a chemical like sodium thioglycolate that combines with and removes oxygen from the media. And that will result in oxygen depletion. At least in the bottom of the thioglycolate tube, there will be no oxygen. That media is usually heated before use to help drive off any absorbed oxygen. And then the sodium thioglycolate chemically combines with any trace elements of oxygen. And in the bottom of the tube, there will be no oxygen. Any question about any of that? We've talked about the thioglycolate medium before, and oxygen does come in to the media from the air, and the tube is uncapped, meaning the cap is loose, so oxygen can come in. So there's actually full oxygen at the top of the tube, and then as you go down the tube, there's less and less oxygen until the bottom of the tube, there's no oxygen. We have selective media also, and this is a media that halts or inhibits the growth of a particular group of microorganisms while not affecting the growth of other organisms. An example of a selective media is one that we have sodium chloride added to it, like 7% sodium chloride media. This is selective for a halophile. Only salt-loving organisms can grow on this media. most organisms, at least most bacteria, cannot grow above 2% sodium chloride. So E. coli cannot grow on 7% sodium chloride media. Any question about any of that? And then one that could be selected for if you were to grow it on sodium chloride media would be Staphylococcus aureus, because the members of the family Staphylococcus can grow in 7% sodium chloride. And as you know, Staphylococcus aureus can be found on the human skin. Everyone has Staphylococcus epidermidis, and the human skin can get up to 20% sodium chloride. Any question about any of that? Another type of culture media you have is differential media. This allows us to see differences between different, like two types of organisms. Usually the differences are visible differences that you can see. The difference is not related to how well the organism can grow on the medium, but because it's visible differences, it's easy to distinguish two different colonies or two different microbes from each other. An example of a differential media is blood agar. Blood agar is differential because you can distinguish between different types of hemolysis. There's gamma hemolysis or no hemolysis, where the blood is unlysed all the way up to the growth of the white bacteria here, and a gamma, that's a Greek lower gamma. You can't see what's happening underneath the bacteria, but up until the bacteria, we can see there is no hemolysis of the bacteria. And then this is alpha hemolysis, the bacteria and the off-white alpha there. And as you can see, let me blow that up a little bit, that right before the alpha, there's a darkening ring. And this darkening of the blood may look dark like that, or it may look greenish in color. And you can see inside the alpha, it's also dark. And that's from the partial hemolysis of the red blood cells. And that differs from this species here in the beta, where the bacteria are streaked in a white beta. It's surrounded by a complete clear region where there's beta hemolysis or complete hemolysis of the red blood cells. And you can actually look through the agar plate right here. I mean, you hold this up to the clock and you could see the clock in this region where the bacteria have totally lysed the red blood cells. It's normal for a beta region, beta hemolysis, to be surrounded by an alpha hemolysis. Any questions about any of that? All right. There are different types of culture media that are combined two or more of the types of media I've discussed, like eosine methylene blue plates, also called EMB plates, are both selective and differential. They're selective, so if something grows on the plate, it's probably gram negative because gram positives are selected against. So gram positives usually do not grow on EMB. It's differential on the basis of lactose fermentation. The bacteria look darkish, like almost black right there, or greenish metallic color, like right there and there, if the bacteria can ferment the sugar lactose. If the bacteria cannot ferment the sugar lactose and they're gram negative, they will grow on the plate, like this one here, and it looks white. Normally the color is the normal color of the bacteria on an EMB plate if it cannot ferment the sugar lactose. And if it's gram positive, it will not grow on the EMB plates. Any question about any of that? Now, if the bacteria cannot grow on the plate, like a gram-positive, will not grow generally on an E and B plate, then you cannot determine the lactose fermentation of that bacteria, meaning you can only determine lactose fermentation if there is some growth on the plate. Any question about any of that? There's also an enrichment media. This is a plate that encourages the growth of a certain type of bacteria and discourages the growth of other bacteria. Therefore, an enrichment media is a selective medium, but we use it differently. We use it to increase the number of the desired microbe to detectable levels. It's particularly useful if the targeted microorganism is only a small proportion of the population. Let's say that you wanted to find a bacteria in soil that could ferment or utilize is it phenol as its only carbon and energy source, meaning a phenol utilizing bacteria. If we were to start streaking out plates to obtain a colony of a phenol digesting bacteria, the chance of getting it would be very remote because probably less than 1 in 1,000 cells in the soil would be capable of metabolizing phenol. In fact, it's probably closer to 1 cell in 1,000,000 in the soil. And so if you were to just go out and start streaking bacteria obtained from the soil, your chance of getting it would be very slim. Because if it is 1 in 1,000,000, you even sample the different 100 colonies, you're not going to find it. But what you could do is take your bacteria isolated from the soil and then grow it in an enrichment media that would select for the growth of a phenol degrading bacteria. And an easy way to do that would be to make phenol the only carbon and energy source in the media. And this would be an enrichment media because it would enrich for phenol metabolizing bacteria. Let me draw this out. So if our bacteria in the soil were 1 in 1,000,000 capable of digesting phenol, we could grow it in a enrichment region media. And let's say it enriches by 100-fold. Then we've lowered that to 1 in, oh, I'm not sure what that would be, 1 in 10 to the fourth. Oop, didn't do that right. That should be a 0 there. Let's go ahead and do that again. So what you could do is put the soil in the enrichment media and now the bacteria, instead of being 1 in 1,000,000, will be 1 in 10,000. And then we can take another enrichment media. Oh, I'm not drawing that tube very well. And take the growth out of this tube, putting it into this tube. And we're just assuming we're enriching it 100-fold. We've now got it down to 1 in 100. So in this tube here, you could then actually streak out the bacteria on a plate, and it's possible to get a colony that would be capable of digesting phenol, because after sequencing 100 colonies or testing 100 different colonies, you would get the one that would be capable of digesting phenol. And it would be even better if we were to do this another time, and let's say it would go only tenfold this time because we're at 1 in 100. And then if it's 1 in 10, you only need to sequence go over here. Just do it that way. This one didn't reach 100-fold because it's so high to begin with. Here, 1 in 100. And 1 in 10, your chance of getting the colony would be very easy. You just need to test 10 different colonies and you get the phenol degrading media. Anyways, this is how you use an enrichment media. Any questions about any of that? All right, this is going through the different types of media that I've just discussed. It's here for a review. Let's move on. A pure culture is a culture that contains one and only one species. If you remember, Louis Pasteur developed a way of obtaining a pure culture, but it was a very difficult way of doing it. And what he did was he used limiting dilution of a broth. And if I still have that there, nope, he would make different dilutions. So we'd have like 1 in 10. I can't do this. 1 in 100. Nope. 1 in 1,000. 1 in 10,000. 1 in 100,000. And of course, 1 in a million as well. And he would take this dilution and then plate it into 10 petri dishes. And if only one, not two dishes, tubes, 1 in 10 tubes, if only 1 in 10 tubes had growth in it, Louis Pasteur correctly surmised that the one tube that had growth in it was a pure culture, and then the 9 tubes that did not have growth in them didn't have a cell plated in that tube. And this one here would, of course, have 10 out of 10. Having growth in it, and he correctly surmised that each of these tubes would be multi-species, meaning it wouldn't be a pure culture. Any question about any of that? Well, Edward Koch came up with a much simpler way of obtaining a pure culture, and that is you streak it out for limiting dilution using the quadrant method here. I'm going to have a hard time streaking this, but you're getting the idea. And then you sterilize the loop, and you should be streaking 20 to 30 times on a sector, and I'm not able to draw that. Sterilize the loop, and then you go back from sector 2 and go into sector 1 to pick up some cells. And you only go into sector 1 about 3 times where the bacteria are low in numbers, and then streak out sector 2 and then sterilize your loop, and then streak out sector 3 going into sector 2 where the bacteria are lower in number. Streaking out about 20 to 30 times, etc. And the idea is that somewhere along the line, either quadrant 1, quadrant 2, quadrant 3, or quadrant 4, we're streaking down single isolated cells so that we get a single isolated colony. And then you pick from that colony cells only from that one isolated colony to streak out a second plate using the quadrant method. And let's say we get an isolated colony right here. And then we take cells from this colony that has gone through 2 colony isolation events. This one right here, which is an isolated colony, and this one right here, which is an isolated colony. And if the cells have gone through 2 colony isolation events, the chance of it being a pure culture is greater than 95%. Any question about any of that? A colony is a population of cells arising from a single cell, or a group of cells, or a clump of cells. And that's why when you get your first colony, about 30% of them will be derived from one or more cells. The colony will then, that cell will then divide mainly by binary fission to grow many times. And that's the colony. You need at least 1 million cells before you can see a colony. We call colonies formed on a plate as a colony forming unit because we do not know if the colony was formed by one cell, 2 cells, or by a clump of cells. And then when we count the colonies on the plate, rather than say there were, I don't know, let's say there's 30 colonies on the plate, instead of saying that this dilution had 30 cells per mil plated on the plate, we say there were 30 colony forming units on that plate. Because like I said, when you count a colony, you don't know if that colony was formed by one cell, 2 cells, or by a clump of cells. And because of that, and there's only 30 colonies, there's more than 30 cells that were plated on that plate. Because some of the colonies would be plated from more than one cell. So obtaining single colonies is critical in obtaining a pure culture. And we do that by using the streak plate method, aka streaking for isolation. And remember, you have to do it 2 times. If you only do it 1 time, you're only 70% pure or about, and that's just not good enough in microbiology. And there I'm showing you the streak plate method. The streak plate method is good when you're isolating a cell that's present in a large numbers in the population. If you were trying to use this method to obtain a cell at very low numbers, like 1 in 1,000 or less, you're not likely to obtain it in a colony for using the streak plate method. Like I said, if the cell was present in only 1 cell in 1,000, then you need to look at 1,000 different colonies. And to try and streak out 1,000 different colonies would be very difficult. You might get 10 to 30 colonies on a plate. Like right there, you got something like 10 or 15. Any question about any of that? All right, let's talk a little bit about bacterial growth. Remember when we're talking about microbes, Growth refers to an increase in the number of cells, not to an increase in the cell size. There are three means by different bacteria to grow. Most bacteria grow by binary fission, just the splitting of one cell into two. But a few bacteria can actually bud, where a parent cell puts out a little bud, and that bud grows and then eventually becomes an independent cell. And then there are also a few species which can grow by reproductive spores, where one cell, let me draw this out, one cell tends to elongate, and then at the tip of the cell, it puts out reproductive spores. The reproductive spores are usually smaller than the parent cell, and they're usually put on one side of the cell. And there's always more than one of them, so that this is reproduction. And what the reproductive spore is just waiting for the wind or an animal to take one of these reproductive spores and take it elsewhere. And then the reproductive spore can go on and form a parent or vegetative cell. This is reproduction because one cell then becomes something like 5 cells, one parent cell and then 4 reproductive spores. That's different from an endospore, which is not reproduction, where an endospore generates, a parent cell generates one endospore inside it. The parent cell then will die as the environment decays, and that will release the endospore. And so we have one parent cell which will die, becoming 0, forming one endospore. So 1 to 1 is not reproduction. Endospores are a survival strategy. A reproductive spore is a reproductive strategy. Any questions about any of that? All right, let's look at binary fission in a little more detail. Binary fission happens when the cell will replicate its DNA. So one DNA chromosome becomes 2 DNA chromosomes, and then they divide where one molecule of DNA moves to one side of the cell, the other moves to the other side of the cell, and then the cell literally starts dividing in two, and it will grow both a cell membrane and a cell wall between the two molecules of DNA, right down the middle. And then the cell will separate, forming 2 daughter cells. Here is actually an electron microscopic image of a cell going through binary fission. Any questions about any of that? Most bacteria do divide by binary fission. A few bacteria divide by budding, where a parent cell here puts out a little bud, which then grows bigger. I guess that's bigger, and that's bigger. And this bud will eventually become the same size as the parent cell, in which case the bud separates from the parent cell. Let's watch a little video of that. I'm not sure how to verify, so I'm glad that did it automatically. So there's the little bud starting to form right there. And this is yeast instead of bacteria, but it buds the same. And there's the bud forming. Oop. Right there, we can see the DNA starting to divide, and it looks like the DNA is starting to move into the bud now. Yep, the DNA is moving into the bud right now. All right, about there. No, I got it in the wrong place. That's hiding it. Right there, this is starting to separate meaning a cell wall and a cell membrane, are starting to form between the parent cell and the bud. It's not totally separated yet, but it will shortly. About right there. And that's totally separated now, where the cell wall and the cell membrane have totally separated out the bud from the parent cell. And that cell is now about the same size as that one. And as they're going on, this cell is going to form a bud here, and that new cell is going to start forming a bud there. Any questions about any of that? All right. When we're talking about cellular growth, it's important to discuss the generation time. The generation time is the time required for a cell to divide, one cell to divide into two, and that's the same time for the population numbers to grow double, to grow into a double size. So the time for one cell to become two cells is the generation time, but the generation time is also the time it takes for a population of 1 million to grow to a population of 2 million, meaning the population number doubles or the cells double. It's the same. The generation time varies considerably between species. It can be as fast as 20 minutes, like E. coli has a generation time of about 20 minutes. Most bacteria have a little slower generation time. So most bacteria, their generation time is between 1 to 3 hours. But Microbacterium leprae is the slowest bacterium I know of to divide. Microbacterium leprae has a generation time of 10 days, meaning instead of the cell taking 20 minutes to divide into two, it takes the cells 10 days for one cell to divide into two. Any question about any of that? Now, when we say the generation time is 20 minutes, it's understood that we mean 20 minutes per generation. So the generation time is the time per generation. So most bacteria have a generation time of 1 to 3 hours. It's understood that I mean the generation time is 1 to 3 hours per generation. And students often get the generation time incorrect because most people think of, I guess, miles per hour. And so they think of it as the number of times the cell divides per hour. That is not the generation time. The generation time is the time per generation. Any question about any of that? We normally plot the generation time on a semi-log plot where the number of generations is a normal arithmetic plot, like 1 generation, 2 generations, 3 generations, 4 generations, 5 generations right there, and then 10, same distance. So this is an arithmetic plot. It's a semi-log plot because instead of having the bacterial numbers in the regular number of cells, we use the log of the number of cells. If we use the generation time and plot it on a semi-log plot, meaning the log number of cells on the y-axis and the generations in the arithmetic axis on the x scale, we note that the generation time is linear. And that's a very easy way to describe the population, meaning the population number increased by y equals mx plus b, a straight line. Another reason for doing that is if you plot the number of points, 1 would be around here, 2 would be around there, 3, 4 would be around there, 8 would be around there, and I don't know if that's 16 or 32 right there. That's 32. Okay? But here, Well, 16 is right there. 8 is right there. 4 would be about there. 2 there, 1 there. Meaning right in here, we have a whole bunch of points, and they're very hard to distinguish. And then the next point here, I guess that would be 200,000, will be on the roof. Can't put it on the graph. But here, the log of 6.2, whatever be the next log, which would be 200,000 cells, it's actually going to go about right here. You can actually put it on the piece of paper. So the semi-log plot is easier because the formula equals y equals mx plus b, very easy. This one is, let me see if I can remember, y equals mx plus mx squared plus b plus c. Or maybe it's c squared, I don't know. A much more complicated formula. And then you can't get the next point on the plot on the arithmetic scale, but you can get it on the graph. You just need to extend the graph right there. And then these points are separated out on the semi-log plot, and they're not separated out on the arithmetic scale. Excuse me here. Oh, some reason I wanted to sneeze there. Hopefully I cleared it. Ah. So that's what we microbiologists like to use the semi-log plot. Any questions about that? All right, let's move on to the phases of growth. A bacterial growth curve often looks like this. And surprisingly, if we were to plot any organism growing, it'll tend to have a growth curve like that, meaning they put a bunch of deer on an island and the deer were transplanted to the island so that they had a lag phase. They explored the island, and while they're exploring the island and getting used to their new environment, they weren't reproducing. And then the deer entered exponential growth phase because there are lots of resources around, only a few deer on the island. And so the deer population went into exponential growth. And then the deer population entered a stationary phase because the lucky deer found some food to eat and then they were reproducing. And the unlucky deer didn't find any food to eat and so they died. And the deer population was stationary at that point, where the number of deer dying equals the number of deer reproducing. And then as the population got older, there's less and less food on the island. And then more and more of the deer started dying because the deer couldn't find enough food to eat. My point is that even deer follow this population growth curve. When you put bacteria from a solid plate into a liquid culture, the bacteria will initially enter a lag phase where the bacteria are not reproducing. What the bacteria are doing is getting the enzymes they need to grow in the liquid culture. Once they have the enzymes they need to grow in the liquid culture, they'll leave the lag phase and enter the log or exponential growth phase, where the bacteria numbers will grow exponentially. Why? Because there's few bacteria in the media, lots of resources, and lots of space. So the bacteria numbers will grow exponentially. But as the population ages and the population number gets bigger, the growth will start slowing. And that's because there will be less space around, more waste products around. Initially, there are no waste products around, and fewer resources around. So the bacteria numbers will start to slow in their growth. And then the bacteria will enter a stationary phase, where if there's a lucky bacteria, it has some resources around it, some space around it, and that bacteria and not too much waste products around it. that bacteria can grow. So that's a lucky bacteria. But there's an unlucky bacteria that will have very little, if any, space around it, lots of waste products around it, and new resources around it, and that bacteria will die. So in the stationary phase, the bacteria reproducing or growing is going to equal the bacteria numbers dying. And that's why the population number does not increase, but it's not decreasing. The population number is stationary, staying about the same. And then as that population ages, there will be less bacteria growing. Only the very, very lucky bacteria will grow. Most of the bacteria will become to die. And late in the death or decline phase, there will be no bacteria growing. All the bacteria will be dying. And this will continue until all the cells are dead. For E. coli, in a week or 2 weeks, all the E. coli will be dead. For something that makes endospores, the endospores can survive for 40,000 years or longer, depending on the endospore, meaning the species making the endospore. The record is they've taken endospores they've obtained from a core of the ice in Antarctica and gotten them to generate, meaning thaw it out and then generate. And I don't remember how long it is. I think it's at least 40,000 years. It might be 40 million years, but something like that. A long time. Any question about the bacterial growth curve? All right. When we're measuring microbial growth, there are a number of methods to measure the growth of microbial populations. There are direct methods and indirect methods. The direct methods directly count the cells in some way. The indirect method will estimate the cell numbers in some way. The direct methods include the viable plate count, also called the plate count, a filtration method, the most probable number method, and the direct microscopic count. We're actually counting the cells. The indirect methods use turbidity, where you're using light to go through a tube to get an estimate of the number of cells. You can get an indirect method by estimating the number of cells from the metabolic activity, like measuring the amount of CO2 being produced. You can also get an estimate of the number of cells by drying the cells and then getting their dry weight. I've used all the methods for obtaining an estimate or the actual number of cells, except for the most probable number. I've never used that. I've used all the other methods. A plate count is the most common method in microbiology for getting the estimate for the number of cells in a solution. It actually measures the viable cells, so this is sometimes called the viable plate count. It and the filtration method are the only ways that measure only the viable cells, meaning a dead cell will not be counted because a dead cell cannot grow into a colony. And what you do is to get your estimate of the plate count is you plate out colonies, count up, incubate the plate, and then count up the colonies. And that's why only a viable cell can give rise to a colony. A dead cell cannot. The results are often reported as number of colony forming units per mil rather than number of cells per mil because we do not know if one cell formed a colony, 2 cells, or a clump of cells gave rise to that colony. Any question about that? So for getting the viable plate count, you first need to make serial dilutions of original sample, and then you inoculate an agar plate with a sample of this dilution. And then you incubate the plates, and you let the bacteria grow, and then you start counting the colonies. So here we're making a dilution. We have our original sample. Take 1 ml of it, put it into 9 mL, and then we have a 1 to 10 dilution. Mix those cells up. Take 1 ml out of that, put it into 9 mL, and now we have a 1 to 100 dilution. Mix these cells up, take 1 ml out, put it into 9 mL, and there we have a 1 to 1,000 dilution, 1 to 10,000 dilution, 1 to 100,000 dilution. And then you take a sample of this. Usually they take either 1 ml or 1/10 of a ml and put it onto a plate. And then you incubate the plate and count the number of colonies. If we use the spread plate method, usually you put 1/10 of a ml on the plate, spread the sample over the plate to get individual cells and clumps on the plate, and then you incubate the plate, and then you count the number of colonies. Using the pour plate, you usually pour 1 ml of sample or the dilution onto the plate, mix it around, and that they're doing a spin plate here. But normally what you do is you put it on the plate. I don't know if you can see this, and then you gently tilt the plate around to spread that 1 ml of liquid all over the surface of the plate. Excuse me here. You then, especially with the pour plate method, allow that liquid to be absorbed into the media, and then you incubate it, and then you count the number of colonies on the different plates. Many of the dilutions will have too many colonies to count. I mean, even this one is too many to count. And these, that's totally too many to count. There, you can almost count no colonies. And you'll get different counts. Now, if this one has 4 colonies on it, and this one has about 44 colonies on it, let's say 34, just to make it a little different. I mean, I could do 40 and 4, but then that would be the same. So let's do 34, and this one will do 4. Which one should you use? You should use a plate that has 25 or more colonies on it to get the estimate of the mean. Because this one, which has 4 colonies, and this one, which has 34 colonies, and what dilution was that? 1 to 100,000. So the dilution was 1 to 100,000. Not very good. Ah. So this would be, we have 4 times 100,000. So that means there's 400,000. Oop. Colony forming units per mil of the original sample. And this one, the estimate is 3.4. Oh, let's not do that way. $3,400,000 colony forming units per mil. And which one is the better one to use? This one is the better one to use because this mean if we have 1 plus or minus 1, the mean here, that's 34. That equals about 34 plus or minus. Is that about 33.3%? I think that's right. Let's make that a minus. I'm not sure how to do an underscore here. Oh, there we go. And this one, let's see if I can just copy that. Nope. So the mean here is going to be 3. Oh, that's, darn, I can't do that. So that's equal 3 plus or minus, get rid of the underscore. 33.3%. Which is a much more variable mean than this mean, plus or minus 3.3%. So we want to use this mean. Any questions about that? All right. Because only a viable cell can give rise to a colony, the plate counts method can only count viable cells. The filtration method is similar to the plate count, but the difference is here we're sending the solution through a filter, and then the cells get stuck on the filter, and then you put the filter on a plate, and then you grow up the colonies. We use the filtration method whenever the cell numbers are very low. Like you could put a whole gallon through the filter, take the filter here and then plate it there. And if there's only a few cells per gallon, you'll get cells growing on the filter that you can count. Any questions about the filtration method? So you're sampling either a solution, a liquid solution, or you can sample the air or some gaseous solution, get the number of cells on the filter, put the filter on an agar plate, allow it to grow colonies, and then count the number of colonies that form. And this would be whatever went through the filter. So this would be, I don't know, that's pretty high, maybe 50 to 60 colonies per gallon if we filtered an entire gallon. Any question about any of that? So you only use the filtration method if your number of cells is very dilute. And obviously it has to be used in either a gas or a liquid. The direct microscopic counts, you can determine the number of cells in a solution by actually counting them under the microscope. You use a specially gridded slide that has a specific volume under it, and then count the cells under the microscope. Here's a specially gridded slide. It has a specific amount of volume under the cover slip. And generally it's designed so that this amount right here under this square here will be 1 microliter. And so the number of cells there, let's say that's 20 cells, you would then say 20 cells per microliter of your original sample. And then that would be 20 times 10 to the 6 because a microliter is 1/6 of a liter. Any question about any of that? Now, the microscopic count isn't good for motile cells because let's say you start counting, you count this row, 1, 2, 3, 4, and then you start counting this row and that cell moves in there. And then this cell, before you get out of it, moves out. So if the cells are motile, the microscopic count is hard to do. If the cells have a large capsule, it's also hard to count the cells. One problem with the microscopic count is you cannot count viable cells, live cells, from dead cells. For example, that cell there, is it alive or dead? Can anyone tell? Generally, you cannot tell a viable cell from a dead cell. The microscopic count, you do require a fairly high concentration, but you could centrifuge down a lower number of cells per mil and get the cells per mil higher. No incubation time is required, meaning you can get the number of cells as soon as the person is done counting, but it's kind of hard on the eyes of the individual doing the counting. Any questions about the direct microscopic count? I'm not going to talk about the most probable number. It uses mathematics and I've never used it, so I'm not going to talk about it. But I will move on to the indirect methods, the turbidity, the metabolic activity, and the dry weight. Turbidity is actually my favorite way of getting a microscopic count. It uses a light going through the media of a tube, and you measure how much light is coming through, and you set that to 100%, the amount of light coming through the media in the tube. And then you get your tube with your cells in it. Let me blow that up. And you'll note that the cells will block some of the light. Some of the light will get through, and the cells will block some of the light. And the amount of light coming through will give you an estimate of the number of cells you have. Initially, you have to have this done and then do a whole bunch of different dilutions, but then you make a chart. So you have like 1 to 100, 1 to 1,000, and then you have this chart or a table. And then you can use that chart or table indefinitely to get your estimate of the number of cells. Any question about any of that? So you're just measuring the amount of light getting through the media, and less light will get through if there's cells in it than if you just have media. So the absorbance is usually reported to get your number of cells per mil. And like I said, you just look at that and then look at the chart and determine how many cells per mil you have. It's very quick. All you do is you put your cells into a tube of media, get a blank tube or empty tube of media, measure the amount of light getting through, measure the amount of light getting through, look it up in the table. One problem with this method is you cannot tell a dead cell from a viable cell. Dead cells will count the same as a viable cell. Junk in the media can also count as a cell. Metabolic activity is where you're measuring the metabolism of some cells and then using that to get an estimate of the number of cells. This assumes that the bacterial metabolic product, like CO2, accumulating is in direct proportion to the number of bacteria present. The first time you have to count the number of cells, measure the amount of CO2 that is given off for a different number of cells, and then make a table of it. And then you have the table, and then you can use that table in the future for indefinite amount of time to determine how many cells you have. You just use the bacterial concentration of unknown cells, measure the amount of CO2, look up at the table and get your estimate of the number of cells. That's how you use metabolic activity. Any question there? Dry weight is a usual method for determining the amount of filamentous bacteria or fungi, where it might be difficult to count them directly because they're strange shape. The organisms can be filtered out of the growth medium. You then dry them, remove the water, and then weigh them. And you can, initially what you do is you count the different number of cells, get their weight, their dry weight, and then you make a table. And then you use this table for all future examples where you have some unknown number of bacteria, dry them, get their weight, look at the table to determine the number of cells you have. The more cells you have, the more dry weight you'll have. One advantage of the dry weight method is usually what you can do is just you centrifuge down your cells, you dry them, and then you can weigh them at your leisure whenever you have time. Doesn't need to be done immediately. A problem with the dry weight is obviously you're going to be counting dead cells and live cells because you can't tell what used to be alive and what's dead. But it's a very easy way of obtaining the number of cells. Any questions about the different ways that we have for obtaining the cell numbers or an estimate of the cell numbers? If not, no questions, that's it for chapter 6. We will begin our next chapter, which I think is chapter 8. Yep, chapter 8. Let me go ahead and save that down. Close it down. Chapter 8 is metabolic genetics. So the genetics of microbes. The major goals of this chapter is know the terms. We'll talk about most of these in just a little bit. 2, be able to describe what DNA replication is, RNA transcription, and protein translation. And 3, understand the very different types of mutation. Any questions about the overview and the major goals of this chapter? All right. Initially, how do I do this? Initially, we're going to talk about the structure and the function of the genetic material. We'll then talk about the flow of genetic information. We'll move into talking about DNA replication, RNA transcription, and protein translation. After that, we'll then change gears and talk about the regulation of bacterial gene expression. How we'll talk about bacterial gene expression is using the operon model. We'll then move on to talking about mutations, talking about genetic transfer and recombination. And lastly, we have a slide discussing genes and evolution. Any questions about Chapter 8? Genetics is the study of what genes are and how they carry information, how that information is expressed, and how genes are replicated. Any question about genetics? A gene is simply a segment of DNA that encodes a functional product. The functional product is usually protein, but it could also be RNA. There are a few genes that only code for RNA, such as the rRNA genes and the tRNA genes. They only code for RNA, no protein is made. Any question about any of that? So here we have a chromosome with the strand of DNA shown here. And as you can see, this is a region of DNA that encodes gene 1, and this is a region of DNA that encodes gene 2. I know this is a eukaryote because there's a region in here that does not code for DNA, meaning there's a region of the DNA that does not code for anything. And eukaryotes actually have a large number, or a large, I think the majority of their DNA is actually non-coding DNA. Bacteria have very little non-coding DNA. Most of the DNA of a bacteria actually does code for a gene. Any questions about any of that? And here we're looking at the X chromosome, showing the different location of different genes. I can't read it, but let's blow it up a little bit. So there's a gene causing muscular dystrophy, but there's actually 2 types right there. Come on. And there's retinitis pigmentosum gene. This whole region might be that one. And then retinitis pigmentosa 2 gene is right there. Any questions about any of that? That's curious. I didn't know that retinitis pigmentosa would be more common in a male than in a female because it's on the X chromosome. Males only have 1 X chromosome. So it should be more common, at least retinitis pigmentosa 2 and 3, in males than in females. Any question about any of that? A genome is all of the genetic material in a cell. When we're talking about microbes, they usually only have only 1 cell, in which case the genome is all the genetic material in the organism. of one cell. The study of genomes is called genomics. A genotype is the genes of an organism. Like this blue-eyed individual has the genotype lowercase b, which gives rise to blue eyes. The phenotype is the expression of these genes. So an individual with genotype lowercase b has blue eyes. The phenotype of an individual with brown eyes is, of course, brown eyes, the phenotype, but the genotype can be uppercase B, lowercase b, or 2 uppercase B, meaning uppercase B, the genotype gives rise to brown eyes, but the uppercase B, lowercase b genotype also gives rise to brown eyes. Now the gene for blue eyes is lowercase b, but the gene for brown eyes is uppercase B. The genotype uppercase B, lowercase b codes for brown eyes, the phenotype, meaning the blue-eyed allele is recessive to the dominant brown-eyed allele. Any question about any of that? Why is that showing? All right. Here we're looking at a chromosome of E. coli. E. coli generally only has one chromosome. Obviously, it's very long, has a lot of genes on it in one segment. Let me roll it up. Like right there, there could be one gene. And the genes are just located on the chromosome, one after the other. Remember, on bacteria, most of the DNA codes for a gene or genes. There's very little non-coding DNA in a bacteria. Remember that the DNA codes for the genes and the non-coding DNA, but a chromosome is both DNA and protein. Any questions about any of that? All right. DNA is a polymer of nucleotides. There are 4 nucleotides in DNA. Adenine, or A, thymine, or T, cytosine, or C, and guanine, or G. You can know either the adenine or the A, but you must know one. I won't require you to know adenine, guanine, cytosine, thymine, but you must know A, T, C, and G, or adenine, thymine, cytosine, and guanine. DNA is a double helix associated with proteins. By double helix, we mean that there's one strand of DNA here that is bound to a second strand of DNA here. The backbone of DNA is the sugar phosphate backbone, meaning this strand of DNA is hooked together from this sugar to the phosphate, from this phosphate to the sugar, in a chemically covalent bond. So a single strand of DNA is held together by a chemical covalent bond, meaning the sugar bound to the phosphate, bound to the sugar. So the sugar phosphate backbone is called the backbone of the DNA, holding a single strand of DNA together. The nucleotide nitrogenous bases of the nucleotide are not holding the DNA, a single strand of DNA together. They're off on the side here. And the bases hydrogen bond to the complementary base in the second strand of DNA. So 2 strands of DNA are held together only by hydrogen bonds between the nitrogen-containing bases. There are two hydrogen bonds between the AT, which are complementary, meaning A always binds with T and T always binds with A. And then there's also the complementary DNA C and G. C always binds with G and G always binds with C. The GC or CG complementary pair have three hydrogen bonds holding the nitrogenous bases together. The two strands of DNA do not run the same. One runs this way and one runs that way, meaning the strands are anti-parallel, one running this way and the other running that way. Any questions about any of that? So remember, two strands of DNA are only held together by hydrogen bonds. The single-stranded DNA is held together by a chemically covalent bond between the sugar and the phosphate. So that means the single strands of DNA are held together much stronger than the double strand, and you could separate the double strand of DNA by simply heating the solution. I'm not sure at what temperature human DNA would totally separate, but at least at 70 degrees Celsius, two strands of DNA will separate and will only be a single strand of DNA. Any question about any of that? All right. Let's talk about the vertical flow of information within cells. This is known as the central dogma, that the information is stored in DNA and that information flows to RNA in transcription, and the information in RNA flows to the information in protein by protein translation. The central dogma of DNA means that the information flows from DNA to RNA to protein. The DNA, the RNA, and the proteins each have their own language for storing the information. Now that's the information flowing within a cell from DNA to RNA to protein, but you can also have the information flowing between generations of cells, which for humans would be between generations, meaning the information starts in the DNA of two parents, and then that information flows to usually one baby. Information flowing between generations of cells or between generations. And the information in DNA flows to two daughter cells by DNA replication, where the DNA molecule divides into two. One DNA moves to one side of the cell, the other DNA molecule moves to the other side of the cell, and then the cell splits down the middle, where both daughter cells get the information from the parent DNA. Or a baby gets its information, its genetic information, from its two parents. Any question about any of that? All right, that's the flow of information between generations of cells. Let's talk about the flow of information between cells of the same generation. You'll note that I've broken this graph, because that's called recombination. What usually happens between cells, the flow of information flowing between cells of the same generation, is the information flows out of one cell. That's called the donor cell. That cell often dies. The DNA is then outside the cell, and then that DNA is taken in by a recombinant cell. And then that recombinant DNA can be expressed and then the cell gains DNA from another cell, such as the original cell, the donor cell. Any questions about that? All right, so the information can be said to flow within a cell, between generations of cells, or between cells of the same generation. We talked about the vertical flow of information within a cell, where DNA stores the information. It flows to RNA, it flows to protein. The language of the information stored in the DNA is the 4 DNA nucleotides. So there are only 4 letters in this alphabet, meaning 4 nucleotides. For RNA, the language is an RNA nucleotides, and there's only 4 letters in the RNA nucleotides. Note that this is an RNA nucleotide and that this is a DNA nucleotide. So the information is slightly different. And then here, the information is stored in the amino acids in a protein, and there are 20 different amino acids, at least in the human proteins. So there are 20 different letters in the alphabet of amino acids. To compare with English, English has 26 letters in their alphabet. So looking at the flow of information, we could say that the information DNA stands for "that is a dog." In RNA, it would be something similar, but a little different, like a different dialect, something like "that is a dog." And then the information in protein would be a totally different language, "quello è un cane." So the same information, but in a different language. All right, so that's the flow of information within a cell. Let's talk a little bit more about the flow of information between generations of cell. This is often called vertical flow of information because the information goes from the parent or the parent cell to the daughter cells or to the offspring. If we're talking about a human baby. DNA flows by DNA replication, where the DNA will be replicated semi-conservative, where one strand will separate from its complementary double strand, and then you use one strand to build a new strand. So this polymerase will bind here, and it'll look at the nucleotide here as a T, and so it'll add an A. And then the polymerase will look at this nucleotide here, see it as a G, so it'll add a C. It'll then move to the next nucleotide, see a C here, and then add a G. And that's how the newly replicated strand of DNA gets made. It looks at the template and then makes the newly replicated strand by adding the complementary base to the base on the template. And that'll happen to both strands of DNA. It's just this one isn't shown. We're only showing it for this one. Any questions about that? So DNA replication will have the polymerase, the DNA polymerase, always add the complementary nucleotide to the new strand that's complementary to the old strand of DNA, which in this case is a G, so we always add the C. By semi-conservative, we mean that if the 2 dark strand or black strands are the original DNA, and the new strand of DNA is the white strand, semi-conservative means that the offspring will always have one original parent strand of DNA and then one new strand of DNA. And that'll be true for both daughter cells, and that we call semi-conservative replication. That differs from conservative replication, where one offspring would have two black strands of DNA, or the parental original, and then the other offspring would have two white new strands of DNA. So conservative DNA replication does not happen. DNA replication is semi-conservative, and don't ask me what the liberal model of DNA replication would be. This is just showing you that the DNA polymerase will bind to this template strand, C and A here, and then add the correct corresponding nucleotide that binds to the T, binds to the A, and that would be the T. And how that happens is it binds a T triphosphate. Two phosphates come off. When that bond breaks, that releases the energy to making this phosphate then bind in a chemical bond to this ribose right there. Any question about any of that? All right, let's watch a little movie on DNA replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any question about that movie? So let's talk a little bit more about DNA replication. DNA is copied by the DNA polymerase. Oop. DNA can only be made in the 5' to 3' direction. It is initiated by an RNA primer, but I'm not going to be quizzing you on the RNA primer, which will then be removed by an exonuclease, and then DNA polymerase or another DNA polymerase fills in what used to be where the RNA primer is. We have two strands of DNA. The leading strand, which is synthesized continuously in the 5' to 3' direction because it's going the correct way. The DNA polymerase must move in the 5' to 3' direction. And then the lagging strand, which is going in the incorrect direction. And so the DNA polymerase will not be able to move down the new lagging strand. What it'll do is will make the strand in short segments going backwards. And these short segments are called Okazaki fragments after Dr. Okazaki, who discovered it. Each time the RNA primer starts the DNA synthesis, but the RNA primers have to be removed and then will be replaced with DNA. The important enzymes to know are helicase. It's the enzyme that opens up the strand of DNA, making it double strand to making it single strand. The second enzyme that comes on is the DNA polymerase. I'm ignoring the enzyme putting on the primer, but the main one will be DNA polymerase, which will read the template strand of DNA and put on the correct base, complementary base to the template base. And then the DNA polymerase will move down to the next nucleotide, read the template strand DNA nucleotide, putting on the correct complementary base for the new DNA strand. So other than the primer, the DNA polymerase could synthesize the leading strand of DNA continuously, and it could even do the entire strand of DNA in one sweep, meaning where the DNA polymerase totally makes the new strand of DNA from the template strand in one go. But oftentimes the DNA polymerase falls off, in which case the process needs to start by another DNA polymerase. And I imagine a primer will be put down first, and then the DNA polymerase will continue it again. So oftentimes that DNA polymerase on the leading strand does not replicate the leading strand continuously in one swoop, but oftentimes that DNA polymerase will fall off, and another DNA polymerase will then bind to the leading strand and finish replicating the leading strand. Any question about any of that? All right. In the lagging strand, as I mentioned, the DNA polymerase cannot work in this direction going down the way the helicase is moving because this strand is running this way in the 5 to 3' direction. And that's the way the DNA polymerase must move. And so what it does is it makes the DNA backwards in Okazaki fragments, short fragments, better than repeated. Like there's one Okazaki fragment, there's one Okazaki fragment, there's one Okazaki fragment. And if you want to, you can say that each Okazaki fragment starts with a short RNA primer. The point is the lagging strand is made in multiple batches, many different batches of Okazaki fragments. And that means the helicase enzyme, the third enzyme of importance, has to work a lot more often on the lagging strand than on the leading strand. Helicase, what it does is, let's say this DNA polymerase began replication right here. Right there, that one was made by another DNA polymerase. So there will be a gap in this newly replicated strand from this part of the newly replicated strand. And let's say this DNA polymerase falls out here. And so that the next DNA polymerase after the primer will be right here. And so once again, right there, there'll be a gap. And the ligase will join this stretch of DNA to this stretch of DNA. And then for this stretch of DNA to that stretch of DNA, the ligase will join them together. The ligase will put in the not the complementary base, the covalent chemical bond linking the single strand of DNA together in the leading strand. The ligase does not have to work very often in the leading strand. If the DNA polymerase were to totally synthesize the leading strand from the template, meaning it would never fall off, ligase may at most have to work one time. And I'm trying to think about that. Does it need to do anything to ligase with the RNA primer? And it might only need to do linking the DNA together where the RNA primer was. But on the lagging strand, for each Okazaki fragment, the ligase will have to work 2 times. It'll have to link this Okazaki fragment to that Okazaki fragment, and it'll have to link this end of this Okazaki fragment to this Okazaki fragment there. So the ligase will be working a lot more on the lagging strand than on the leading strand. Anyways, those are the 3 enzymes that are important in DNA replication. The helicase, making the double strand DNA single strand. The DNA polymerase, using the template DNA to make newly replicated strand of DNA. And then the ligase, joining the newly replicated DNA strand together, where there are different batches of the DNA, like in each Okazaki fragment. Any question about any of that? All right. I think that's it. So in DNA, all of the DNA gets replicated. I think before talking about transcription, I'm going to ask, are there any questions? And if not, I'm going to just stop here because this is a good place to stop. And we'll talk about RNA transcription next time. All right, if there's no questions, I'll say goodbye and I'll see you at 7 in the lab. Bye.